ती कुठून मिळेल
ग्रुपवर येते नाही आहे का तिकडे
ग्रुपवर व्हॉट्सअपला आहे युट्यूबला जाते ना तिथून दिसेल मला का लाईव्ह केलं तू स्टार्ट करून मला काही स्क्रीनशॉटच घ्यायचे हो थँक्यू
So is my presentation visible or is it not?
visible
Okay.
वेलकम टू स्लाइड आपण डायरेक्ट सरांची
Yes. So, good afternoon, everyone. My name is Shauna Kulkarni, and I'm from Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research. And welcome to one-day national webinar on exploring innovations in novel drug delivery system under the aegis of Internal Quality Assurance Cell (IQAC). And it is organized by Gokhale Education Society, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nashik. Welcome, everyone. So the next speaker for today is Dr. Ashish Parashar. He's a professor and head of Pharmaceutics Department in Lloyd Institute of Management and Technology, Greater Noida, Uttar Pradesh. And the topic sir will be covering is nano-theranostics, shrinking the gap between diagnosis and treatment. And to tell you something about Sir, he's a PhD from Oriental College of Pharmacy and Research, Oriental University, Indore, Madhya Pradesh. He's a Master of Pharmacy specializing in pharmaceutics from the Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur University. And he's also a Bachelor of Pharmacy from Department of Pharmaceutical Sciences from Dr. Hari Singh Gaur University. And currently, he's professor and head of the Pharmaceutics at Lloyd Institute of Management and Technology, Greater Noida, Uttar Pradesh. And to tell you something about his research, he has guided over 15 MPharm dissertations, specifically in F&D or formulation and development and drug delivery. He has reviewed papers from many internal journals. He's organized many international seminars, conferences, and symposiums by esteemed institutions like AICT, ICMR, DPT. He is LOC member of committee under member IPA Convention 2014. Additionally, he has a lot of publications. He has research papers, he has review papers, and also he has 4 book chapters. Along with this, Sir has 4 patents. And so over to you for today's presentation. Thank you, sir, for being with us.
Okay. Thank you very much for this nice and wonderful introduction of mine. And before starting my topic, my presentation, I would like to thank the entire team of this webinar, organizing team of this webinar, especially the principal, Dr. Sunil Amritkar sir, and Vice Principal, Dr. Prashant Pingle sir. Uh, Participating in webinar and giving a lecture is really a great opportunity, and for this opportunity I am very thankful to all of you. Dr. Prashant Pingle sir, he's my senior, and he's like my elder brother, and many times when we met, he guided me what to do and how to excel in the field of pharmaceutical science. So Prashant sir, thank you very much for being there always. Now I'm going to start today's this lecture on nano-theranostics, shrinking the gap between diagnosis and treatment. I, Dr. Ashish Kumar Parashar, I am currently working as a professor of pharmaceutics in Lloyd Institute of Management and Technology, Greater Noida. So I am also thankful for my institute for providing me the opportunity to participate in such a beautiful programs. I would like to welcome all of you to the world of nanotheranostics, where cutting edge technology is revolutionizing the medical diagnosis and treatment. So in this lecture, in my presentation, I will explore you the fascinating field of nanotheranostics, its potential to transform healthcare. It is very novel term and very new field to work. It is only around 20 years back. So it is like a very open field to work, and if somebody is interested to work in nano particulate drug delivery carriers, then he can choose nanotheranostics as their topic. Now, few students, when I talk them about the nanoparticles or nanotechnology, they say, "Sir, this is very new field." Comparative to nanotheranostics, nanoparticle is or nanotechnology based carriers, it is a very old field. For your information only, I am talking about this. Why it is very old? So I must say that we Indians are using these nano carriers from since very long time period. And I am giving you one example. I think you people may agree. What happens in my times? Currently, I don't know what the mothers they practice or not. But at my childhood time, my mother used to put a kala tika over my head, and that kazal also in she used to put in my eyes. Whenever I asked why you are putting this kajal over my head or in my eyes, she always told, this will help you to say this will save you from the bad eyes, बुरी नजर से आपको बचा के रखेगा. My dear friends, why I am saying all this? Do you know what is that kajal or that kajal contains? You will surprised to know that that kajal contains carbon nanoparticles and carbon nanotubes. So we are using this thing since very long. This is not new for us. But in my lecture today, I am going to talk about nanotheranostics. A little bit a different term. I think most of the people, most of the pharmacy professionals, they are not aware of this. My presentation will contain the introduction, what are nanotheranostics, how they work, their applications, challenges associated with the nanotheranostics, their future, and the conclusion. So, starting with the introduction, I would like to draw your attention. Sir, yes. So I would like to draw your attention towards the diagnosis first. We are the pharmacy professional, and we are responsible to develop new formulations, new drugs. What about diagnosis? After all, we are working for the treatment of diseases. If you are not having true idea about the disease, then how can we deliver the drug to that particular site, or that particular, how can we target that particular disease? So my dear friends, I have included a term, that a sentence, the poor diagnosis may result in inefficient treatment. If doctor is not having the right idea about the disease, then there are very little chances that you will get good result of treatment. First, I will explain this thing by a general example, and then we talk about the specific examples. There are some diseases in which doctor may get confused, and if proper diagnosis is not there, then doctor cannot give you the right treatment. For example, jaundice is there, and malaria is there, and sometimes hepatitis also. So in all these cases, what happens? Our RBCs, they are degrading fast. Its degradation is fast. And if proper diagnosis is not there, then doctor may get confused in between the jaundice and the malaria and some other symptoms. And in India is a very so developing country. It's having a very limited resources, limited treatment availability in say uh, remote areas. So in those areas, what happens? Doctor, he prescribe medicines on his assumptions only. Now it's your luck. Maybe possible your disease will be cured, and maybe possible it will be worse. Then you have to move to some big cities. So, friends, diagnosis is very important for the efficient treatment of diseases. And now I take you with the cases, the severe diseases like cancer. If you are not having the true idea or the correct idea about the disease, what kind of cancer is there, where the cancer cells are locating, what is the prognosis of the disease, whether the stage is metastatic or it is not, it's just limited to some particular site. If proper diagnosis is not there, then treatment of cancer is somewhat difficult. And yes, I would like to mention that diagnosis is there, still the treatment of cancer is very difficult. Why? Because of the severe side effects associated with the cancer treatment. In India, we are having only two, three types of treatment like surgery, followed by chemotherapy and radiotherapy. Therapy. That's it. However, rest of the world in developed country, immunotherapy and targeted therapy is also there. In India, it is started, but it is quite costly, and general population is not able to afford it. So, what will be better? It will be better if we get idea of the severe disease in early stages. And similarly, there are some other diseases also where theranostics can be used, like uh, atherosclerosis, diabetes, or Alzheimer, Parkinson's. So I will discuss all this in later slides. So what I focus and what I think, that if we are having the correct idea about the disease, the treatment can be the better treatment can be given to the patient. And the same I have written in the slide. A shorter gap between diagnosis and treatment typically result in improved patient outcome, especially for cancer patients. If the conditions that are left untreated or are diagnosed late can progress to more severe stages, and making it difficult to manage, potentially reducing the chances of full recovery. So, if diagnosis is proper, treatment can be given on time and patient can be cured. And if diagnosis is not proper or you are having a late diagnosis of disease, severe, I am talking about the severe diseases, then they are also called as life-threatening diseases. Okay? And there are chances that patient may get died. I would also like to mention this importance by making you familiar with COVID or Corona. In COVID period, what happened? If somebody is affecting from Corona or COVID, they used to go to the government laboratories. Sample was collected, and the report was coming later after 4 hours or 24 hours. And during this period, patient was, he feared of what happened with me. And in the COVID time period, not only the patient but the entire family that was threatened. So diagnosis is very important, my dear friends. Now, what are the say challenges associated with the traditional diagnosis? They are time consuming. Yeah, as I have given you the idea that in time of corona, the report was coming after 24 hours, and patient was struggling to say for MRI and CT scan whether they are affected, how much their lungs are affected, and the report was coming after 4 hours, 8 hours, 24 hours. And remote areas, the condition was very worst because they are not having a proper diagnostic arrangement. So the first drawback I say that the current diagnostic strategies they are time consuming and they are highly dependent on the symptoms. If somebody is having some symptom, then only you can diagnose. Otherwise, who will go to the doctor? Nobody will go. But in some cases, like diabetes, in case of hypertension, in case of say cancer, suddenly patient come to know that he is suffering from this disease. Sometimes patient they avoid, थोड़ा बहुत कुछ हुआ they avoid. Oh, it's okay, कुछ नहीं, कुछ खाने पीने से हो गया होगा. So next is invasive procedures. In many cases, for the diagnostic test, they require invasive procedures like biopsy, surgical interventions, which are painful, risky, and may lead some complication. For example, if somebody is having lung cancer, so it is a very difficult to take the sample from lungs. Next, yes, it is costly. Costly in case when you have to go for frequent diagnosis. For example, again, I will talk about the cancer. In case of cancer, we have to go for frequent CT, MRI, and sometimes we have to go for PET scanning, positron emission tomography, and this is very costly. So if you go for MRI, you go for PET scanning, then my friends, it's very costly to locate the disease, to find the disease, its condition. And in case of cancer, it is required that the frequent testing is there. Sample requirement is there, and mishandling of sample is also there. In some cases, blood is taken as a sample, or urine is taken as a sample, or tissue is taken as a sample. And due to some mishappening, the person, the pathologist, mishandling of pathologist, inaccurate result may come. This is also another problem associated with traditional diagnosis. And it is also inconvenient. Inconvenient for the patient where fast results are required, accurate results are required, assumptions are not required. Okay, so now what we can do or what scientists are doing? Scientists, they have thoroughly learned this thing, and then they have started working on a new area that is known as theranostics. Theranostics, the term was given by John Fackhauser, US consultant, in August 1998. So you can see this is a very new field. It involves a set of diagnosis, a drug carrier, and the observation of treatment reactions. You can see the word theranostic. It is made up of merging two different words, therapy and diagnostics. Now I think you can get idea that the same molecule which we are going to use as a diagnostic agent could act as a therapeutic agent also. And this is the basic funda behind theranostics. Theranostics, these are the devices, offer the advantages of concurrent diagnosis and treatment for certain diseases like cancer. Now, in certain diseases, we also say everybody is curious about what is drug doing inside the body, as you have seen in previous lecture, how the aerosols they are moving inside the body, lungs, air pipe, then lungs and alveoli, how they are working. This we can't observe, but using theranostic, it is possible now. We can observe what the treatment strategy, what the treatment or the drug is doing inside the body, and where it is acting, and how much it is acting. So, monitoring of the biodistribution and function of the administered therapeutic agent and detection of target tissue can be simultaneously tracked using theranostic nanomaterial. Simultaneously, we can see whether where the carrier is moving or where the drug is moving and what drug is doing in that area. That is also important. Whether the drug is attacking the disease cell or not, we must know. So, again, this theranostic field is vast, and I am focusing on nano-theranostics, nanomaterials used as theranostics. What are nano-theranostics? Nanotheranostics is an emerging field of medicine that combines the diagnostic and therapeutic capabilities of nanotechnology. Now, again, new word. Therapy is there, diagnosis is there, but including nanotechnology. Okay? Now, unlike theranostics, which focus on imaging and image-guided therapy, nanotheranostics, they involve using nanoparticles to both diagnosis and treat diseases at molecular level. It means, my dear friends, we can see where my drug is moving, which cells are interacting with my carrier, how my carrier is acting inside the body, how my carrier is attacking the affected cells, and how my carrier is curing the disease. All we can see after injecting these nanotheranostics inside the body, and we can observe using external device what is happening inside the body. Nanotheranostics, these are the class of innovative nanoparticles that combine diagnostic agent, therapeutic agent at a single platform. They are nanocarrier, they are having a therapeutic agent like some drug, DNA, RNA, and they are also having diagnostic agent with them. Now, these teeny wonders are engineered to enable targeted drug delivery. A very important term, targeted drug delivery. Why targeted? Because in certain cases like cancer or Alzheimer, where some particular cells they are affected only and not entire body, we need to attack only those cells which are affected by some disease. So in such cases, targeting is very important. I will explore how nanotheranostics they target the disease. So targeting is one aspect, and another is real-time monitoring of the treatment response. After delivery of nanotheranostics, after reaching these theranostic agent to the targeted site, what happens with the cells? So this can be observed by some external device, and this may also help us to work for personalized medicines. Why we need personalized medicines? This I'll explore you. So say, how many nanosystems are there which can be used as nanotheranostics? We don't have to work on some novel things. We have to work on the available nanocarriers. We can work on metallic nanoparticles. We can work on polymeric nanoparticles, solid lipid nanoparticles, micelles, NLCs, liposomes, dendrimers. We don't have to work on nanocarriers. We just have to use the available nanocarriers, which are currently researched and well established. For example, liposomes and nanoparticles, they are well established. So these nanocarriers, they combine with some therapeutic agent. It may be chemotherapeutic drug, nucleic acid, protein, or stimuli responsive some molecule. And another is imaging agent like quantum dots, gold nanoparticles, gadolinium based contrasting agent. So by combining nanocarriers, therapeutic agent, and imaging agent along with targeting agents, we can develop systems. Now, nanoparticles. One by one, we will discuss different components of nanotheranostics. So, first part is nanoparticles. I have included the term nanoparticle. It means it includes all nanocarriers. Okay. So, nanotheranostics involves the use of nanoparticles for both diagnostic and therapeutic purpose, and their size ranges from 1 nanometer to 100 nanometers. They can be engineered to fulfill some specific, say they can use to achieve some specific targets. For example, targeting, drug targeting. They can be made biocompatible. They are having property of control release, as you have I think you may aware of control release systems. So nanoparticles, nanocarriers, they are having inherent control release property. And these properties makes nanocarriers ideal candidate for use as nanotheranostics. By modifying their surfaces, their surface property, because one of the most important limitation of nanocarriers is their surface charge and surface properties. And because of their surface charge and surface properties, they are identified by the reticuloendothelial system in body, and RES uptake is there. One question in your mind can arise that, sir, we can use simply nanoparticle. Why we prepare nanotheranostics? So this is the reason. Nanoparticles, they are identified by the reticuloendothelial system. RES uptake is there, and we are not able to get 100% result of nanocarriers. So after surface modification of these nanocarriers with some biocompatible material like PEG, polyethylene glycol, or sialic acid is also there, and some other moieties are also there, but mostly we use PEG, pegylation. It is well established and marketed formulation is also available. So attaching PEG over the surface of nanocarriers, it helps to stealth the carrier from RES. It helps us to reduce the RES uptake of our nanocarriers. So surface modifications is there. These are different kinds of nanocarriers. You see, magnetic nanoparticles can be used as nanotheranostics, niosomes, nanospheres, micelles, nanocapsules, nanoshells, solid lipid nanoparticles, dendrimers, gold nanoparticles, liposomes, quantum dots. Any of these carriers can be used as nanotheranostics. But the question must arise, sir, if the carrier is hollow, then drug and imaging agent can be entrapped. But what about the solid particles where the drug and the carrier will be allocated? So in those cases, we can attach our imaging agent and drug over the surface of nanocarrier by taking help of this PEG. This also includes the surface modification. Okay. Now, friends, second component of nanotheranostic agents is targeting agent. Targeting agent, these are also known as targeting molecules or ligands, and these are the substance used to specifically interact with or to bind to particular cell, tissue, or molecules. They are having certain property by which they bind only to certain molecules or certain cells in body. They will not interact with all the other body cells. They will interact with particular cells for which they are engineered. These agents are designed to enhance the precision and selectivity. Yes, they help. In case of cancer, I'm talking more about cancer because this is my field of work. So, in case of cancer, we want to target specifically the cancer cell and not the normal cell. And for this, we cover up our carrier, our nano carrier, using this targeting ligands which attack only cancer cell, which interact with cancer cells only and not normal cells. What happens? I will tell you. I explain this. For example, what happens in cancer? Cancer, a simple definition is uncontrolled cell growth, cell development, or cell division. Uncontrolled cell division. And for this uncontrolled cell division, when the cells they are frequently dividing, they need nutritious material more comparative to normal cells. More nutrition they require, like glucose, like amino acids. So we use these material as a ligand. We cover up our, we modify the surface of nanocarrier using these kinds of material which cancer cell require as a ligand, and then we can target the cancer cells. There are some example of targeting agent which are currently in use: antibodies, monoclonal antibody, and their fragments. They are used as targeting molecules. Peptides, amino acids, as I have told you, and aptamers. Aptamers are short, single-stranded DNA or RNA molecules that can bind to specific targets with high affinity. So, first we have selected the nanocarrier, then we have modified its surface with some, a neutral biocompatible material, and this biocompatible material and encapsulated carrier, or say this kind of biocompatible carrier, can be attached with targeting ligand. Targeting ligand will attach over the surface of nanocarrier. How it works? You can see. This nanoparticle containing drug inside, this is a liposome, it contains the drug inside and it is circulating with the blood. It is having a specific type of ligands over its surface, and whenever these ligand finds the suitable receptor to bind, they escape from the blood circulation, reaches to the tissue, and they bind specifically with their loving receptors. And when ligand receptor binding is there, this carrier, the entire carrier, is engulfed by the cell. And these ligands, they facilitate the movement of the carrier inside the cell. And after entry into the cell, this carrier reaches to the lysosomes or endosomes, and there they are digested and they release the drug. And in this way, they target a specific cell only. Now, next important component in nano-theranostic is imaging agents. Imaging agents, also known as contrast agents or tracer. These are the substances used in medical imaging. They are already in use to enhance the visibility and diagnostic accuracy of a specific tissue, organ, or physiological processes within the body. They are already in use. And what they do? They help us to find the area where the disease is concentrated, and they also give us a improved contrast between the targeted area and the surrounding tissue. So exact visualization of disease can be done. Different types of imaging agents are there. Radiography, some agents they are used in radiography and CT scanning, like iodine and barium. Some agents, contrast agents, they are used in MRI, magnetic resonance imaging, like gadolinium. Few nuclear tracer are also used. They are radioisotopes, radiopharmaceuticals, and they can be detected by positron emission tomography or single photon emission CT, computed tomography. So PET or SPECT, in both of these techniques, we used radiopharmaceuticals. And some fluorescent dyes also available. However, in nanotheranostics, we focus less on fluorescent dye because they are having less time to work in body and they are degraded fast. So these all are existing imaging agents which we can use to entrap in nanotheranostics. We don't have to work on imaging agents also. So now you can say that nanotheranostics, these are just the combination or compilation of these three different techniques: imaging agents, targeting agents, nanocarriers. Now see, we are having these different imaging agents. I have shown two carriers, imaging agents. First one is quantum dots. These are the nano substance. They are semiconductor, and according to their size, they emit different color of light. You can see the size is small, they are emitting green light, and if the size is increasing, their color is changing, and it changes from green to yellow, then orange, red, and then maroon. Similarly, we can also use FDG. It is fluoro deoxydoglucose. And fluorine, here what we use? Radioactive fluorine. Fluorine 18 is used. However, you can also use oxygen, carbon, nitrogen, radioactive carbon, radioactive oxygen, radioactive nitrogen, but their half-life is very small. So in PET scanning specifically, which is running in India and in entire world for the detection of cancer, so in PET scanning, mostly 18F FDG is used. It is having around 2 hours of half-life, so it can be used. And it gives the result, it concentrates to the particular site in the cancer cells only and give idea where cancer is existing in the body. Now come to the next level, that how these nanotheranostics works. So first we have to formulate these nanotheranostics. We have to select one carrier. We synthesize that carrier and then encapsulate the drug and the imaging agent, or we can simultaneously develop the carrier, entrap the therapeutic agent, and entrap and or encapsulate the imaging agent simultaneously. So both approach can be used. Then these carriers, they can be surface modified, or during synthesis, during synthesis also they can be modified. Their surface can be modified, and then they are bind with the ligands which we choose, and this depends on the type of disease where we want to target. So depending on the target site, depending on the receptors, we choose the ligands. So we formulate, we develop a nanocarrier, encapsulate drug, encapsulate imaging agent, then surface modification, then ligand attachment over the surface. This all we have to follow to develop a nano-theranostic agent. Now, nano-theranostic agent, they are formulated, they are developed in lab. Next stage, we must know how they will work, how and what they will do inside the body. So after formulation, see, I have one video is there to show how nanocarriers work. So first, I think you will see, and then I will explain how they will work. This video is taken from Australian universities.
Scientists are using cells isolated from human tumors to develop new ways to tackle cancer. Here we can see the cancer cells growing in the culture dish in the laboratory. For most drugs to be active, they must go inside the cancer cell. Scientists are developing tiny particles, nanoparticles, that can be loaded with drugs. The nanoparticles can be modified so that they attach to the cancer cell and get carried inside. To make sure that the drug-loaded nanoparticles only attack the disease cells, they are engineered so that they only bind to the target cancer cells and not to neighboring healthy cells. Once they bind to the cancer cell, they are taken into the cell. They can't pass through the surface. Instead, part of the cell surface forms a pit and then it engulfs the nanoparticle. Now we are looking inside the cell. Cellular proteins wrap around the vesicle and drive it inside the cell. You can now see a vesicle carrying the nanoparticle moving towards the very center of the cell. The vesicle carrying the nanoparticle travels further inside the cell and then fuses with a compartment called an endosome. This is part of the digestive system of the cell's stomach. The inside is acidic, and its job is to digest incoming material. The nanoparticles are degraded inside the endosome, releasing the drug into the cell. The drug can then kill the cancer cell. Nanoparticle-based drug delivery provides a way of attacking cancer without the side effects of conventional chemotherapy.
So I think, my dear friends, you have got some idea how nanoparticles they work, how they reach inside the body, they reach to the targeted site, then they attach with the receptors with the help of ligand receptor binding, and after that they get entered into the cell where they reaches to the endosomes and maybe lysosomes, and then there they are digested and they release their drug. And simultaneously, in case of nanotheranostics, when they release drug, they also release the imaging agents, which can be detected from outside the body using different imaging techniques. So now we need imaging techniques. Here are few examples of imaging techniques like magnetic resonance imaging, MRI, highly used in India, CT scanning, PET scanning, SPECT, TIRF, total internal reflection fluorescence microscopy, or in some cases near infrared emission for bioimaging. Now, in the figure I have shown here, you can see using normal MRI, normal PET scanning, we can get idea where is the disease is located, but not complete idea how much is it has spread it. While using nanotheranostics agents, we can specifically find how many cells they are affected by the disease. And at each level, we can increase the resolution of these techniques, and we can find where our carrier is moving and where the disease is existing. Now, one question may arise in your mind. Sir, PET scanning is also there, already there, MRI, CT is already there, then why we go for nanotheranostics? So here comes the main point that yes, we have scanned that cancer is lying in lungs, or cancer is there in brain, or cancer is there in breast. This we have detected using MRI or PET or SPECT or anything. But now what we are doing, we are using nanotheranostics to simultaneously delivery of drug. So during the diagnosis, we are delivering the drug, we are targeting the cells, we are delivering the drug, we are killing the cancer cells during the diagnosis. While currently what happens? Doctor, they write first go to biopsy, and after biopsy, if it confirmed, then PET scanning is there, some other test, MRI, PET, CT, and then they start treatment, writing prescription, or they are start chemotherapy or surgery, anything, or radiotherapy. And again, the problem arises with current treatment strategies. What happens? After looking the PET report or MRI report or biopsy, they start chemotherapy. Now, doctor don't know where this chemotherapy going. Either it is going to the targeted cells or it is going to entire body. So, with current treatments, what happens? Chemotherapeutic agents, they move into entire body and they show high number of side effects. Severe, in some cases, severe side effects. Yes. For example, hair loss is there, nausea, vomiting, patient lose appetite, he don't want to eat anything, and suddenly you will see a heavy weight loss is there. Then chemotherapy is continued. Slowly, different organs, they start failing. Retinopathy, nephropathy, neuropathy. Different side effects are there with the associated with the chemotherapy when they are non-targeted, and they affect normal cells as well as cancer cells. So nanotheranostics or targeted nanocarriers, they are solving one problem, that they will target only cancer cells and they will leave normal cells. Okay. But still, with targeted therapy, you have to first go for diagnosis and then targeted therapy, then diagnosis, then targeted therapy, then diagnosis, then targeted therapy. So what will happen? Expenditure is very high, cost will very high. And countries like India, it is not possible to spend this much of money. So nanotheranostics may help there. Now, I would like to tell some advantages. Key benefit is targeting. Then, second is simultaneous imaging is there. Now, what this simultaneous imaging will help? This will help for the potential. This is having the potential for personalized treatment option. Now, what is this? For example, some patient is suffering from lung cancer, and initially the cancer size is around 3 millimeter or 2 millimeter. He needs some dose of drug. Then after chemotherapy, the size is reduced. So the dose should also reduced. But in case of conventional chemotherapy, it is not possible, because chemotherapy, it runs in cycles, different cycles, and in one cycle dose is constant. Now using nanotheranostics, when we are getting idea that disease is shrinking, cancer size is shrinking, so we can reduce the dose. Yes. So this is the difference. We can develop carriers as per the requirement of patient and as per the requirement of disease. If disease is shrinking, then we can change the treatment dose. And what will be the effect of all this? This will help us to get improved outcomes of treatment and reduce the risk of adverse reactions. Additionally, nanotheranostics can provide real-time monitoring of the treatment response. Yes, this is also very important in case of some diseases like cancer, or say atherosclerosis or Alzheimer, where our nanotheranostics carrier, what we will do, they are giving real-time images that the disease is shrinking or not. Now let me tell you, in case of cancer, conventional chemotherapy is there. But what happens? After few days, patient, they do not respond to the treatment. Why? Because of drug resistance. Okay. Now, drug resistance is there, and you are prescribing, doctor is prescribing conventional chemotherapy, patient is taking overdoses of chemotherapeutic or cytotoxic drugs, and what will happen? It will result in very high adverse effects, severe adverse effects. So, if real-time monitoring is there, we can also find that our drug is working or not, our carrier is working or not, our treatment is working or not. And if our treatment is not working, then we can modify the treatment. So this is also a big advantage of nanotheranostics carriers. Now talking about different diseases where this nanocarriers, they are applied, or their application is in different diseases. So first one is cancer, deadliest disease of the era. Very, very dangerous disease. And why I am saying this? Because if somebody is suffering from cancer, then only and only around 1% chances are there that the patient will survive. Otherwise, using conventional chemotherapy, surgery, or radiotherapy, patient cannot be survive for more than 5 years. And in some cases, in some cancers, this time is very less, 8 months, 10 months, 12 months. For example, brain glioma, survival time is only 10 months, 12 months. In most cases, patient may get dead. Very dangerous disease. Why it is dangerous? Because uncontrolled growth is there. Uncontrolled cell division is there inside the body, in any part. I'm saying any part, it means it may be breast, lung, liver, kidney, brain, eyes, anywhere, skin, anywhere. Now uncontrolled cell division is there. Number of cells, they are increasing very fast. And what happens? They start affecting the function of their nearby organs, nearby tissues. They start destroying their nearby tissues, and then organ failures occurs. Most of the patients, they fear of cancer. Most of the population, they fear of cancer. Not because of this, this is the deadliest disease, but it is all, its treatment is also very costly. Comparative to other diseases, the treatment of cancer is very costly. लोगों के घर जमीन जायदाद बिक जाती है. This is a very dangerous disease with having a very huge expenditure. And that's why many of the people, they fear of cancer. कि अरे भाई किसी को ना हो. Now I want to say yes, people fear of cancer, but yes, cancer survivors are also there. If you are getting treatment timely, if you are getting the treatment on time, patient can save their life. You can see Manisha Koirala, Yuvraj Singh, Sonali Bendre, all these, they were affected from different kinds of cancer. They fight with the cancer and they won the battle. But this is not the case of common man. This is the case of our heroes. Common population, they do not have that much of money that they go to USA and that get targeted treatment or immunotherapy. It's quite costly. Targeted treatment is very costly. So, if scientists could succeed in nanotheranostics field, I hope that this could be done in India also, and we can save many lives, many patients who are suffering from cancer. Now, what happens in case of cancer? The big problem associated with the cancer is cancer stem cells. Very smart cells, very advanced cells, and very active cells. They are very say cunning cells. Cancer stem cells. See, when doctor when I was talking about the cancer, suppose the case is breast cancer. What doctor do? They remove the breast, and now patient he thinks that he will be safe from cancer. No. After surgery, they need to take chemotherapy, and in some cases radiotherapy also. Now, can we say patient is saved from cancer? No. There are still chances that after few days, few months, or later on, patient will again affected by some other type of cancer. And this is the real story. This is the real story. If somebody is suffering from some type of cancer, after few days, you will see that he is also suffering from some other type of cancer. And in case of untreated cancer, you can find metastatic stage 4 cancer, where treatment is very difficult. So cancer stem cells, I was talking about cancer stem cells. These are the types of cells which escape themselves during the chemotherapy. During conventional chemotherapy, these cancer stem cells, they are having capability to save themselves from the effect of chemotherapeutic agents or drug. They escape. And as soon they find favorable environment, they again start growing. And this is the challenge. if cancer stem cells are there and if cancer stem cells are surviving, then after a short time period, it may be 6 months or 8 months or 1 year or 2 year, patient will again diagnose with some type of cancer. So using nanotheranostics, we are aiming to target cancer stem cells also, so that complete eradication of cancer could be done. Okay. Now, one another problem with the cancer is drug resistance. Yes, my dear friends, after 2 or 3 cycles of chemotherapy, our body cells, cancer cells, they start resisting the entry of chemotherapy therapeutic agent inside the cells. Now, if drug is not going to enter inside the cell, then what will happen? Chemotherapeutic agent, they will kill normal cells and they will leave cancerous cells. No, any development will be there. No betterment will be there. And my dear friends, this is also one of the practical problem associated with the chemotherapy. Yes, I have actually done the survey and I have collected the data from different hospitals from India. Like I would like to acknowledge the Sri Aurobindo Institute of Medical Sciences Indore. I would like to acknowledge Rajiv Gandhi Cancer Research Center, New Delhi. I would like to acknowledge the Tata Memorial Hospital, Mumbai and Varanasi, the KIMS Hospital, Hyderabad, Jawahar Cancer Hospital, Bhopal. I have studied the cases of cancer running in these hospitals and what the problem doctors facing and what the problem patient facing there. And I came to a conclusion that drug resistance, if occur in any cancer patient, then you cannot treat that patient and his or her death is, that is the final destination. So again, our nanotheranostics agents, they can help. How they can help? I have told you, personalized medicines, personalization of the treatment. When we see that the chemotherapy is not working or our nanotheranostics agent is not giving the response, disease is not showing any response, then we can change the ligand, we can change the chemotherapeutic drug, we can change the we can use the combination of chemotherapeutic agent, and we can also use the combination of ligands, not single ligand, 2 or 3 different ligands. So after doing all these modifications, there are chances that the drug resistant case may start responding. Okay. So in case of cancer, nanotheranostics, they offer promising approach to cancer treatment by combining diagnostic capabilities, imaging, their potential for targeted drug delivery. They involve nanocarriers. So yes, it is a benefit of nanotheranostics agents that the damage to healthy tissue can be avoided. Otherwise, you can see as soon the chemotherapy starts, hair loss is there, visual disturbance is there, chakkar bhi aata hai many times patient ko. So, and in some cases, you can find the appetite, loss of appetite, nausea, vomiting, किसी काम में मन न लगना. There are many side effects associated. But using nanotheranostics, we can avoid the damage to the healthy tissue. We can only target the cancerous cells. Second is, in addition to targeted drug delivery, nanotheranostics also allow imaging. So yes, Now we can say, yes, my drug is working on cancer only. It is not affecting the normal cells. It is not affecting the hair cells, hair follicles. Yes, with nano-theranostic agents, we can assure that our carrier is working on cancer only or the disease site only, not the other sites. And in this way, we can reduce the side effects as well as we can get better treatment, better cure of disease. And personalization is also this personalized medicine. This is, somewhat the need of the hour. Currently, what happens? Single type of medicine is given to all type of patients. Same dose, same medicine given to all types of patients. Whether we never see whether the dose should be reduced or increased or what to do. And in case of chemotherapy, yes, doctor, they used to reduce the dose or they increase the dose depending on the status of cancer. But during the cycle, the dose remains same. Okay, and due to this only, side effects are there. Now, I would like to show you how nano-theranostic agents work. I have shown you in previous slide how the nanocarriers target the cancer. In this video, you will see how the nano-theranostic agents may help to kill the cancer specifically. So see this. So see how the theranostic agents, they are migrating from blood circulation to the affected site, means cancer site. They specifically going to the cancer cells. Then using external device like MRI or CT scan, we can observe what is happening with the cancer cells. Now see, here we have located the cancer cells and using some external source of energy, we stimulate our theranostic agents. The temperature is increased and cancer cells, they are killed. Now see the future. If we work with theranostic agents, what we will achieve? Nano robotics. So in this way, in future, I hope we will be able to fight the diseases. I would like to acknowledge the University of Exeter for creating this video. So friends, I think in this previous video, you have got some idea that if we are having targeted nano-theranostic agent, it will reach to the target cells only, and then either using external device or using some internal medicine, we can kill the cancer cells specifically, avoiding normal healthy cells. Okay. Now, apart from cancer, where these agents can be used? So yes, you can see the next disease where nano-theranostic agent can be used are cardiovascular diseases. Here you can see if somebody is affected from atherosclerosis, where the plaque is deposited inside the blood vessels. Using these nanotheranostic agents, we can remove these plaques. Again, what will be happen? What happened? These nanotheranostic agents will reach to the targeted site with the circulation, blood circulation. They will bind with the plaque, and they will release certain chemicals, certain drugs, which will help to reduce the size of plaque, and after some time this plaque could be removed. So scientists are also working to develop nanotheranostic agents to fight with atherosclerotic plaques and to cure heart diseases. Another example is neurological disorder. Alzheimer disease is there, Parkinson is there. Now, with the ability to cross blood-brain barrier, very important thing. Normal drugs, many of the drugs, they cannot cross blood-brain barrier. A tight junction is there, proton GP, EGP pump is there, which effluxes the drugs from the brain. So, nanotheranostics, when they are having attached with the ligands, specific ligands which can cross blood-brain barrier, they can help to treat neurological disorders. For example, nanotheranostic agents which are they are having ligands, say NGO-PAP. NGO-PAP is a category of ligand which are specifically used to target brain. So yes, and there are many more ligands, but here I am talking about NGO-PAP. It is specifically used for brain targeting. So what we do, we attach NGO-PAP over the surface of nanotheranostic agent and then deliver inside the body, and it is having capability to cross blood-brain barrier very efficiently. So using nanotheranostic agent, we can cross the blood-brain barrier and reach to the brain. Now the next stage. Again, in some cases, we can use such ligand which attach with the damaged cells, neurons only, and not the normal neurons. For example, in case of Alzheimer, damaged neurons, our nanotheranostic agent may attach to the damaged neurons and may deliver drug or nourishment to those neurons and may treat the disease. And similarly, they can also be used in case of Parkinson. However, using nanotheranostic agents in these diseases, it seems a little bit costly. A cheaper treatments are available. So now, come, what nanotheranostic agents are doing? Nanotheranostics bridging the gap between diagnosis and treatment. What they are doing? Timely intervention. Early diagnosis allows for timely intervention. Doctors may get idea about the disease and they get to plan the treatment. What treatment should be given? So, using nanotheranostics, doctor in very small time after diagnosis of the disease. One more thing I want to clear you, that in case of cancer and certain diseases, direct nanotheranostics are not used. Theranostics, they are used only after establishment of the cancer. Say after diagnosis, proper diagnosis, like biopsy or PET or MRI or CT. After this only, the role of nanotheranostics starts. Okay, but what nanotheranostics helps? It helps to reduce the frequency of diagnosis, because after each chemotherapeutic cycle you have to go for diagnosis. But using nanotheranostics, once you have started nanotheranostics, you don't need to go again and again for diagnosis. Once you inject the formulation, you can see where the formulation is going and what is happening. Enhance the quality of life. Bridging the gap between diagnosis and treatment can significantly enhance the patient quality of life. Symptoms can manage, discomfort or pain can be alleviated. Okay? Cost saving. Yes, when frequent diagnosis is not required, then yes, cost will be saved. For example, I have told you that in a PET scanning, one time expenditure is around 20,000 to 30,000. One diagnostic cost of PET is around 20 to 30,000 rupees. So if you are using nanotheranostics during the treatment cycle, you will find you don't need to go separately for a diagnosis. Then prevention of complications. Yes, during the disease, some severe complication may arise. Using nanotheranostics, we can see if there is any severe complication, and we can also start treating those. For example, in case of diabetes, kidney associated diseases or vision problems or neuronal degeneration is there. So using nanotheranostics, we can avoid these things. And advancement in medicines. Yes, with growing science, with the growing field of artificial intelligence, with the growing field of robotics, we also have to make our medicines smart and advanced. So yes, we need to work over this to make the medicines smart. For this, many scientists they are using robotics. Many scientists they are using artificial intelligence, and in future, I hope we'll be able to see these kinds of treatment common in our cities. Now, there are certain challenges associated with the nanotheranostics where we have to work still. One of the major challenges is developing nanotheranostics. Developing nanotheranostics is navigating the complex regulatory landscape. Say, I have told you we have to develop the nanocarrier, we encapsulate drug, encapsulate imaging agent, then surface modification, binding of ligand. Do you think this is very easy? This is making of samosa? Put the aloo inside the envelope and develop samosa? No, my dear friends. There are huge challenges with the nanotechnology. Yes. First, that the reproducibility of these nanocarriers is a big limitation. And scientists they are working on such techniques so that nanocarriers could be reproducible. Okay. Second is stability problem. The same problem which liposomes or nanoparticles encountered, the same problems are associated with the nanotheranostics. Next problem is regulatory aspects, FDA guidelines, approval processes. However, scientists, researchers, they are working on it. They are trying to get approvals from US FDA and the other regulatory agencies for experiments. Because if we are not getting the permission for human studies, then this will be like a literature only. This will not come into the market. Another challenge is manufacturing, which I have told you. It is a little bit expensive. Why it is expensive? Because development of nanocarrier is expensive. Chemotherapeutic agent, they are itself expensive. Then ligands, they are having high cost. And then their manufacturing environment, aseptic area, control of the methods, mechanisms, techniques. So they are comparative to currently available conventional chemotherapies, nanotheranostics will be costly, but as they are more effective in long term, they may be cheaper. Okay. Another problem associated with these carriers is quality control and their stability in clinical settings. Till date, many scientists, and including myself, we have developed nanotheranostics in laboratory. I have developed nanotheranostics agents. It was quantum dots encapsulated PPI dendrimers, which I worked on during my PhD work, and I have developed. But during animal studies, I faced challenges. Yes, dear friends, results were not prominent, that much prominent, and it may be due to non-availability of the facilities also. We are in India, we are having very limited facility for research. So at that time, I didn't got very promising result with my career, but it may be possible with some other career like liposomes or solid lipid nanoparticle or polymeric nanoparticles. Result may be more say beneficial or good. Now, future of nanotheranostics. If we continue working in labs and if we get all facilities, then this is the future of nanotheranostics. Advanced nanomaterials. Ongoing research aims to develop new nanotheranostics with enhanced performance, with better targeting capabilities and improved biocompatibility. Yes, my dear friend, one more challenge is there when we are going to use solid nanoparticles, metallic nanoparticles, or quantum dots. They are themselves toxic, and their movement from the body, it is not easy. They are themselves toxic. So studies are continue with these. Gold nanoparticles are there, silver nanoparticles are there, but scientists are still working on their compatibility with the body. Next is integration with medical devices. Yes, my dear friends, some scientists they are also working on integration of nanotheranostics with some devices like pumps, implants, catheters, so that once these devices are put inside the body, they could work for years. Whenever cancer is there, the device may sense certain chemicals in blood and they automatically release the nanotheranostics agent. But this doesn't have done till date. It is proposed. And nanorobotics, I think many of you have heard this term, nanorobotics. So, nanorobotics, it is still in development phase, where scientists they are trying to develop such carriers, such say drugs, carriers, or molecules which act as robot, and they will repair the cell or disease site, as I have shown you in previous video, that these nanorobots they find the damaged DNA or RNA or any damaged cell inside the body, and they will reach to the site, they will repair the cell or the DNA or the RNA, and they avoid the malfunctioning of those cells. So nanotheranostics is also working in this field. Now I would like to discuss few case studies where nanotheranostics were used. One study conducted by researchers at University of California, San Diego, they used nanotheranostics to improve the effectiveness of chemotherapy for ovarian cancer. And after using nanotheranostics, 80% of the patient they respond positively. Okay, so this is just due to the target specific drug delivery. Similarly, some study published in journal Nature Communication, where nanotherapeutic agents they were used for the treatment of Alzheimer, and they have also found some promising results. However, this field is in growing, is just development stage. It is not well established, and still we need to work more and more on this field. So, with this, I would like to conclude my topic, my lecture, and in conclusion, I can say nanotherapeutic has the potential to revolutionize the healthcare by offering personalized treatment options, enhanced imaging capability, and improved drug delivery using targeting capabilities. The use of nanoparticles in this field allows the targeted therapies that minimize side effects and improve patient outcomes. Okay. Then, although there are challenges associated with the developing and implementation of nanotherapeutic, including regulatory hurdles, a collaboration between academia, industry, and government can help to overcome these obstacles. The only thing which we can do in India is start working on nanotherapeutic agents. In most of the Indian institutes, nanoparticles are formulated. Either liposomes, niosomes, dendrimers, nanoparticles, anything. There are synthesizing. What we have, what we need, we just go forward and move forward to convert these nanoparticles into nanotheranostics agents. Just we need to incorporate imaging agents also. This we can do in India, and as soon we start this, we can also start move forward for the development of and establishment of nanotheranostics in our country. With continued research and development, nanotheranostics has the potential to transform the way we approach disease diagnosis and treatment. These are the references I have used in my presentation. I have also put the references along with the figures wherever they are mentioned. And lastly, I would like to acknowledge the organizing team. I am immensely thankful to Dr. Prashant Pingle sir, Dr. Sunil sir, for inviting me and providing me the opportunity to present my talk on nanotheranostics in this wonderful webinar. I would also like to extend my thanks to Mr. Anmesh, who has coordinated with me to conduct this webinar. And also, I would like to thank the entire team of organizing team of this webinar. In addition, I would like to thank Dr. Vandana Arora Sethi, Group Director of my home institute. She always encouraged me and all other faculty members to take part in such activities and share our views, our knowledge with the budding pharmacy professionals. I would also like to thank everyone who is for taking the time to learn about the exciting field of nanotheranostics. You can also see my contact. You can find my contact. I have mentioned my email ID as well as the mobile number. Whenever you want to work, if you are having some will to work in this field, then I can help you how to design the proposal, how to work in this field. So thank you everyone. Thank you organizers for calling me. If you are having any questions, you can ask.
Thank you, sir, for this wonderful talk. And so, actually, we have two questions. I'll be displaying it to you. You might see them on your screen. So, sir, we have a question, and the question is that can we use theranostics in nano emulsions?
Actually, with nano emulsions, we are having many limitations. Nano emulsion itself is an unstable system. Okay, and when we want to formulate nanotheranostics, we have to use different techniques. We have to say chemical modification, attachment of ligand over the surface of carrier. So it is a little bit difficult for nano emulsions. I don't think that it will be feasible with nano emulsions, and there are chances if we formulate this, then there are chances the formulation will be highly unstable. So I don't think that we must go with nano emulsions.
Okay, sir, thank you. And, sir, we have another question. Recently in the UK came forward with Tecentriq drug, which they said is effective against cancer. What do you think about it?
Actually, day by day, if we see many drugs, they are coming into the market, and say immunotherapy. In case of immunotherapy, many drugs are there. They are effective against the cancer, but I have told you that we are facing two problems. First one is the cancer stem cells, which are having capability to modify themselves. Okay, so most of the chemotherapeutic agents they fails after some time. You can find huge number of chemotherapeutic agents are there, large number of chemotherapeutic agents. My dear friend, I have interacted with a doctor. I cannot tell his name. He came back from USA. He was working 10 years there in USA hospital, cancer hospital in USA. And in one case, that doctor, he belongs to KIMS Hyderabad. He surprisingly he told now there is no treatment for a particular patient. I was talking about a patient. Why this patient is not responding to the chemotherapeutic agents, radiotherapy or immunotherapy. Why is not responding? Then that doctor told me we have tried each and everything. Now drug resistance is there, and this patient cannot be now he cannot be treated. He or she cannot be treated. So my dear friends, drugs are there, huge number of drugs are there, but drug resistance is there. Okay. Now, if you are you're talking about Tecentriq, some drug, it will take around 10 to 20 years for this drug to come into the market. And if it is successful, I wish that this drug should be successful. But dear friends, due to cancer stem cells and due to drug resistance, most of the chemotherapeutic agents, they are not working well. And we have to change our strategy. Either we have to move on some nanotherapeutic like research, or we have to move some Ayurvedic treatment, because using chemotherapy, I am very sad to say that in most of the cases, patient could not survive for more than 5 years. So I think no more questions are there.
Yes, sir. No more questions. Sir, thank you very much for your valuable time and your insights on nanotheranostics, its challenges, its advantages. Thank you very much, sir. I'll now forward my screen to Borase, sir, who will be giving the vote of thanks. Yes, sir, just a minute. Sir, I'd like to humbly request you to accept this certificate as a token of appreciation to you. Thank you very much for delivering your lecture, sir.
Thank you very much. Thank you, everyone.
Thank you. Now, sir, I'll be giving the screen to Borase, sir. He'll be giving the vote of thanks.
Okay.
Thank you, Shonak. So, good afternoon, everyone. On behalf of Cochlear Education Society's Dr. M.S. Gosavi College of Pharmaceutical Education and Research, I am Mr. S.S. Boroste, coordinator of this one-day national level webinar, feels privileged to propose vote of thanks. It has been truly enriching experience, and I would like to extend my heartfelt thanks to the participants who made this event possible and successful. First and foremost, I would like to express our sincere gratitude to our distinguished speakers, Ms. Neetu Saudagar and Dr. Ashish Parashar sir, who generously shared their expertise, insights, and knowledge with us. Your presentations were not only informative but also inspirational. Today, I would like to remember our late Sir Dr. M.S. Gosavi sir, former Secretary and Director General, Cochlear Education Society. Sir, Sir, your blessings and inspiration are always with us. I would like to offer sincere thanks to Dr. Mrs. D.P. Deshpande, Madam, Secretary and Director HR, Cochlear Education Society for continuous support and motivation. I am grateful to Dr. R.P. Deshpande, sir, Joint Treasurer and Trustee, Cochlear Education Society for his valuable inputs. I am thankful to our mentor, Mr. S.M. Gosavi, sir, Establishment Director, and Mr. P.M. Deshpande, sir, Project Director of Cochlear Education Society, for their moral support and making facilities available for today's webinar. I must mention our deep sense of appreciation to our principal, Dr. S.V. Amrutkar, sir, for his constant guidance and support. I would love to acknowledge the hard work and dedication of the organizing committee for this well-structured event. And once again, thank you all. We look forward for more such enriching experiences in future. Thank you so much. Thank you, sir.